move right on to Stephanie Telesca's presentation. And I met Stephanie, or we connected, I think, about a year ago after her daughter was diagnosed with another potassium channelopathy in the KCNH1 gene. And she and I quickly became very close friends because we're dealing with this crazy thing of trying to raise severely disabled kids while also starting a foundation and raising money and trying to develop treatments. So she's been a wonderful support to me. But the reason she's here today is because she actually was a part of this rare disease world long before her daughter was born in a professional capacity. She is a genetic counselor, so I thought she would be the perfect person to explain the basics of the genetics of KCNH1 to all of us non-scientists on the meeting today.
Thank you, Michelle, for the introduction and for inviting me to speak today. It is a great pleasure to be here to speak to all the families and help give you the tools to understand a little bit more about your child's diagnosis and how to navigate this new world, and also giving you the tools to help understand the path forward with the treatment options. So I'm just going to pull up my presentation. Feel free to ask questions as we go along. However, if you want to wait until the end, that is also appropriate. As I mentioned, this is really just for you guys to learn the basics. So we will be talking about, just genetics 101. This is, our overview will start very broad. We'll talk in general about DNA, genes, chromosomes, talking about different inheritance patterns, what variants mean and what the classifications mean. And then we'll talk a little bit more specifically about ion channels and how the variants can impact the ion channel and how that will influence our approach to treatment. So just a little bit about me. I graduated from Sarah Lawrence College Human Genetics program in 2013. It's a Master's of Science, and soon after that became an American board certified genetic counselor. I've been working in the field of medical genetics since then and currently hold a role in prenatal genetics at Mount Sinai Hospital in Toronto, Canada. But I met Michelle, as she said, about a year ago after my daughter, Eliana, was diagnosed with a KCNH1 gene variant, so a different potassium channel, but very similar in terms of the outcomes. I've also established a foundation for my daughter's condition to help raise money and awareness to accelerate the path to a cure. All right, so if we take a step back and think about high school biology, you may all recall that our bodies are made out of billions and billions of cells. Inside each of our cells, we have over 20,000 genes or instructions that tell our body how to grow and how to develop. All of these genes are packaged into structures that we call chromosomes. And if you were to take one chromosome and unwind it, you'll see that it's made from a very long strand of DNA. Segments or pieces of our DNA, we call them genes, and genes have their own alphabet of A, T, G, and C. So these different combination of these four letters create different words that our body can read through, and then it creates proteins, and proteins are what are doing work in our body. When we think about genetic conditions can happen at the level of a chromosome. It can happen at the level of the DNA strand, but today we're going to focus on genetic conditions, or one genetic condition that happens when there is a typo inside of the gene, so having a spelling mistake inside that gene. Before we talk about those typos, I'm just going to focus on where KCNH1 gene is and show you that when we take all the chromosomes from one cell and create a picture, we have something called a karyotype. And we all typically have 46 chromosomes that form 23 pairs. The first 22 pairs of chromosomes are the same in both males and females, and it's the 23rd pair that differ. Females have 2 X's and males have 1 X and 1 Y, which I don't have a an image of. But the KCNH1 gene is located on chromosome 1 in this little area here at 32.2. We have 2 copies of the KCNH1 gene, and typically they are both working just fine. So just, and this is just important to remember when we talk about the inheritance patterns. So a lot of people who have a child with KCNH1 may not have a family history of this. And this is because this condition can happen by chance, or what we call de novo, and this is something that happens at the time of conception. There are genetic conditions that can be inherited from an affected parent, and then not related to KCNH1, but there are conditions that you may hear of that happen because the parents are carriers for the recessive condition. So that means that both the copy from mom and the copy from dad, are not working properly. But in KCNH1, it's just one of the 2 copies of that gene that are non-functional that cause the disorder. So in these cases, when it's not inherited from an affected parent, we call this a de novo dominant. So both mom and dad are unaffected. And when they have a child, they see that one of the kids may have the spontaneous mutation or variant that causes the diagnosis. So I'm just going to, okay, sorry, I was looking at the chat. You may also have heard of some dominant conditions that are inherited from an affected parent. So we call this autosomal dominant because it does not matter if it's from the mom or the dad, and they can pass on the condition equally to a son or a daughter. So in this condition, the affected parent will either pass on the genetic condition that they have 50% of the time, or the normal working copy of that gene 50% of the time, and have an unaffected child. And then rarely, there can be a case of what we call mosaicism. So mosaicism happens after conception, when there's a genetic change that happens in one cell of the embryo. And as that one cell divides and grows into the baby or adult, parts of the individual will be affected by the genetic change or the genetic condition, and the other parts are not. So this individual would be called mosaic. So they don't have 100% of their cells that are affected by the genetic condition. And when someone who is mosaic goes to have a child of their own, they may have up to a 50% chance of passing that on. It really depends on where that, what cells in their body have the genetic change. And if it's in their gonads, so the eggs or the sperm, then they will have a 50% chance of passing that on. And their child would be 100% affected. So sometimes this can happen where a child is diagnosed with a genetic condition. It could be KCNH1. And then when the parents have follow-up testing, they find out that they actually have the genetic condition as well. And they may not have the same type of severity of symptoms as their child because not 100% of their cells in their body are affected. And this is just to show with recessive or autosomal recessive conditions, when both mom and dad are a carrier for a recessive condition, they have a 25% chance of having a child with the genetic condition and a 75% chance to have an unaffected child. Again, we don't see this with KCNH1 families, but I just wanted to bring this to your attention as you may hear about this when you're doing research and looking into different types of treatment modalities. So what are variants? And when people talk about variants or mutations, what is this referring to? So variants are, normal to have. We all have them. They make us who we are. So we all share almost like over 99% of the same DNA, but it's these small changes that make us unique, make us who we are. Variants can be classified as benign, unknown, or pathogenic, and the guidelines on how to determine what a variant is or what category it is classified into is based on a guideline from the American College of Medical Genetics. It incorporates published reports, computer programs or in silico predictions, empiric data, and functional studies, and sometimes even segregation studies. So how is this variant being inherited in a family, and who has it and who doesn't? Benign variants are also thought of as normal variants. So this is when they have information that says the variant is not responsible for causing disease. And then you may also hear of likely benign, and that means that there's a greater than 90% chance that the variant is not responsible for causing the disease. And an example that I like to use with the benign variant is the word color, where if you are American and you spell color, you spell it C-O-L-O-R. And if you are Canadian or British, you spell color with a U, so C-O-L-O-U-R. When you read them, it is a different spelling of the word, but it means the same thing, and it is not considered a spelling mistake. Variants of uncertain significance are just variants that are found that have insufficient data about that variant in order to know if it's responsible for the diagnosis or the disease or not. You know, we all, these variants that we have in our genes, we don't always understand how it's impacting our body. And that is because no one is like a native DNA speaker. We don't have a reference to go to say, is this a normal variant? So a U or not a U in the word color, or is it a full typo, meaning that it's really not the way to spell that word? With more research and data that is collected over time, variants of uncertain significance are typically reclassified. And then pathogenic, which is what I think a lot of families here would find on their child's genetic test report. A pathogenic variant means that that variant is responsible for causing the disease. And then you may also see likely pathogenic. And again, that's a greater than 90% chance that the variant is responsible for causing the disease. And an example of this is if you were to remove the letter L from the word color, it does not mean the same thing anymore. And you would look at that and say, well, I'm not really sure what they're trying to say. So it would be a complete spelling error. Okay, so now we're going to jump into ion channels, and this is just an image of a human cell. We're not really going to focus on all of that. We're just really going to look at the plasma membrane, and that's where we're going to find these ion channels that we're going to talk about. So an ion channel is found, as I mentioned, in the plasma membrane, and this is, it allows ions or charged molecules to either enter the cell when it's open, or they can exit the cell membrane. And this is one way that cells communicate with one another. We know that with the cell membranes, they all have like a neutral charge. So if we think about like electrical activity, and this is how we can think about sometimes the ions moving is causing the cells to become positive or negative, or more positive or more negative. But they all sort of start at this like neutral charge, and the flow of the ions into and out of the cell will change that. And that is one type of communication that we see. And this type of communication is represented by what is called an action potential. I'm not going to go into all the details about this, but essentially when the cell receives a signal, it will depolarize and you'll have an influx of ions, and then it will then repolarize where there will be a flow of ions out, and then it comes back to its neutral or its resting state so that it can begin the communication all over again. Okay, sorry, I missed Lynn's question. So the risk of having another affected child, we can get, it really depends on if the condition was inherited or not. So Lynn, I'll respond to you separately. And if other people have questions, I can maybe go into a little bit more detail about that at the end. Sorry. So When we are thinking of KCNH1, it is considered a voltage-gated potassium ion channel. And this allows potassium to flow out of the cell when it opens. And what it's really doing is it's bringing back the cell to its neutral state or resting state. And this allows, as I mentioned, for the continued flow of information. And this is just an image of a neuron. So KCNH1, it codes, or the instructions of that gene create the potassium voltage gated channel KV10.1. So a lot of people may refer to it as KV10.1 rather than KCNH1, or you might hear both of them together when referring to the disorder. This channel is found in the brain, the heart, and it's also identified in a lot of different cancers or tumors. Disease-causing or pathogenic variants in this gene are associated, with different human diseases, and specifically the syndromes that are associated with are the Temple-Barretzer syndrome or Zimmerman-Leban syndrome type 1. They're considered neurodevelopmental disorders and developmental and epileptic encephalopathies. So this is just sort of like the umbrella condition it falls under. When the ion channel or KV10.1 does not work properly, and this is because of the type of variants that are in the gene, and the variants will cause the gene to do different things. So you may hear of loss of function, and this can sometimes cause a reduction in the number of channels that are present, or cause the channel to open too slowly or close too fast. So then you don't have enough potassium leaving the cell membrane or the cell, and the cell cannot continue communicating at the right speed to the next cell. I think a lot of individuals with KCNH1 related disorders will have a gain of function, and this causes the channel to open too quickly, and it does not close fast enough. And so you can think of this as being like a loose channel. So too much potassium is leaving the cell. And again, this does not allow the cell to continue communicating at the right speed. So in summary, the electrical activity of the brain cell is disturbed when the ion channel is not working properly, and that can lead to an increased risk of seizures or like abnormal electrical activity. The disruption of the proper brain cell function also impairs normal function and therefore normal or typical development. So this is all important to know because our approach to treatment, although the goal is going to be the same, which is to normalize the flow of ions into and out of the cell, knowing how the variant is impacting KV10.1 will allow us to approach it appropriately. So when we are talking about a medication, we want to know, do we need to close the channel faster, or do we need to leave it open longer? So that would depend on whether or not it's a gain or a loss of function. There's also protein regulation, and this is when we try to reduce the abnormal KCNH1 protein from being made. So then you're either going to need to upregulate the normal copy or not. And so this can improve the way that the KV10.1 channel is working, because if you have less abnormal copy, then you're going to have the channel that's made properly. And then in terms of gene editing, this would be targeting this specific typo in the gene and going in and fixing it. And I think this is something that we all aim towards, but right now it's not something that I think is necessarily in the works. So to summarize all the information, KCNH1 is an autosomal dominant neurodevelopmental and epileptic encephalopathy disorder. A majority of the cases are de novo. However, there are some individuals that have an affected parent. KV10.1 is the voltage-gated potassium channel that is coded by KCNH1 gene. Pathogenic and likely pathogenic variants impact how the flow of potassium leaves the cell membrane. And treatment modalities are dependent on how the flow of potassium is impacted.